Hello and welcome to the Swine Disease Reporting System. This is the report number 54. My name is Edison Magalhães here at Iowa State University.
Hello, I am Giovanni Trevisan here at Iowa State University.
Hello, my name is Guilherme César from Iowa State University.
And today we're going to cover the findings from the previous month, the month of July 2022, at the Sdrs report. And also we're going to have a discussion with Dr. Marcelo Almeida. He's our invited guest here today at the Sdrs. Dr. Almeida is currently a clinical assistant professor at Iowa State University, serving as a diagnostician in the veterinary diagnostic laboratory. Dr. Almeida got his DVN from the Universidade de Brasília and his PhD in population sciences animal health here at Iowa State University. His PhD was on PER surveillance in weaning age piglets, and currently Dr. Almeida is investigating APP and E. coli outbreaks in the US. So Dr. Almeida, it's a pleasure for us to have you here today and looking forward for the discussion. Thank you.
Thank you all guys for having me here today.
So yeah, let's get started.
So thank you, Dr. Almeida, for accepting our invitation to be part of our show. And before we start our discussion with Dr. Almeida, Giovanni, what are the main findings in the Sdrs for the month of July?
Well, we are in the time of the year that we usually expect to see less activity of the agents in the field. And when we think about that, the only exception that we have is Mycoplasma pneumoniae that start growing up the detection at this time of the year, and that's according to the expected and is occurring. On the other side, if you look specifically for PERS virus, there is this decreasing activity of PERS virus during June, July that's happening at an overall level. But when you look at the regional level, there still be activity of this virus above expected in states like Missouri, Nebraska, South Dakota. Those are associated with more detection of the lineage 1C variant strain. So it seems like the strain entered in the beginning of the year was a lower detection during March, April, and then in May and June start to have a new wave of increased detection of this strain there. So this is consistent with what we see in the field. And some reporting of depopulation of breeding herds has been sent to us by the advisory group, and it seems that that's going to help to decrease the detection of this agent. On other regions like Indiana, example, we still have increased the activity of PERS virus, and this is due to a different strain from a 174 lineage 1A strain that is happening in that region and causing problem in the field. For other agents like PD has been a hot topic this year with lots of detections going on in the beginning of the year. We know that there was a regional spread and it is current, the detection is still above expected, most in the Corn Belt region, but else in North Carolina. However, when we see that there is a decrease in detection by PCR of this agent and looking specifically for a state ICU diagnosis that the number of tissue cases are going down, that's a good signal on the horizon that this may be under control. If you look for other agent that is caught our attention is influenza A virus, and specifically detection from the winter-marked age category. The month of July historically has been the month that with lower overall detection for this agent. But this year we saw an increased detection occurring in the winter-marked age category. So there is some reports of some cases of flu happening in some of those finishing barn that is atypical the presentation. That may be a contribution for this increased detection. Aligned with that increase for PCV2, we saw really high detection occurring in winter-marked in the month of June that's consistent with July. So something is going on that could be more surveillance or some clinical presentation of this agent that's occurring. It's not clear yet. And from confirmed tissue diagnosis at Iowa State, we had some cases of salmonella that pick up in some weeks. Even though it's very limited number of cases, there was some mulberry heart disease. And the feedback that we receive from the advisory group is that this is consistent with what we see in the field, and that is this continued detection of E. coli in nurseries that is occurring over time, and still there this problem. Also, some reports of some case of clinical presentation of elites occurring in the field. So this is a high-level overview of what's happening in the field and lots of different activity across different agents.
Oh, very good. Thanks, Giovanni, for the updates from the previous month. And now we're going to move forward to our conversation with Dr. Almeida. So at Dr. Almeida, we're going to focus on outbreak investigation and population-based sampling discussion. But Dr. Almeida, as Giovanni just mentioned, for example, advisory group mentioning about the E. coli in the field in nurseries predisposing to co-infections with other pathogens. So you're currently working with E. coli strains that affect the industry, causing losses in sow farms and both sow farms and nurseries. Could you give us an overall situation of E. coli in the U.S. and what are you investigating regarding this pathogen?
Yeah, sure. Very good question, Edson. So in the conversations that I've had with different production systems, different vets across the U.S., I think the perception is that we have an increased number of cases, especially post-weaning due to E. coli. So when we look at the data generated here at the lab, which does not represent, a prevalence at the United States, but it can give us some trends of what's going on over time. It's very clear that we're having an increased number of cases of post-weaning colibacillosis. So just to give you some numbers, for example, from 2011 to 2018, we had an average of diagnosed cases of 326 cases per year. And then in 2019 and 20, we averaged about 540 cases, so we had an increase in there. But in the last year, that was 162 or 862 confirmed cases of post-weaning E. coli. So that's almost 60% increase considering the numbers from 2019 and 20. So that's a big jump. So due to that increase, we kind of started investigating some of those cases along with some vets in the field. And with the help of Dr. Ganwuli here, we did some whole genome sequencing of some strains. And what he found was this new Digen gene, which is present in cases of human consequence or diarrhea in children, but had never been reported in sick pigs. And that gene is called TIA. And what we're doing now, thanks to some funding from the ASV Foundation and the Iowa Livestock Advisory Council, is to investigate the role of that gene in post-weaning diarrhea, if that's contributing to more severe disease, and also what's the effect on if the current products that we have available for controlling E. coli can actually control isolates that contain that gene or not. So that's what we've been seeing, and that's what we're working on in terms of research with these new E. coli strains.
No, great.
Dr. Almeida, just a complementary question. Just like if we are having or facing some E. coli cases in the farms, which kind of samples should we send to get the results and also to isolate and have more information about these strains?
So I guess in general, just for enteric challenges, the best samples are samples from pigs that were acutely affected, non-medicated, that you're euthanizing when you are collecting those pigs at the farm, because then we'll have the chance for better preserved tissues for both the histopathological evaluation, but also for culture. And then, again, you submit that fresh and refrigerated and also fixed, and we'd like at least one section of colon. Make sure you include one section of ileum and 4 or 5 sections representative of the rest of the small intestines, and that would give us a good ability to detect whatever challenge might be present in those pigs.
Oh, great.
Those are great information, Marcelo, and thank you for sharing that with the Swine Disease Reporting System audience. That's very much appreciated. And you have been out of the grid doing this type of investigation about emerging or re-emerging pathogens. And if we recall correct, it was at the end of 2021 when we started to see some increased tissue diagnosis of APP, and you went further with that and started to further exploring and doing some field-applied study in this area of what was going on, a regional area problem that was happening in Iowa, more specifically. Mm-hmm. Would you mind sharing with us what you did learn with that, and what is the take-home for the audience about this APP situation that was going on here in Iowa at the end of 2021?
Yeah, I think first we would just like to acknowledge SCHIC for funding a few initiatives to investigate what happened there with APP. And some of our collaborators in that project, which include Dr. Daryl Holtkamp, Dr. Gustavo Silva, Isadora Machado, Mafada Mil Homens, Ana Paula Silva, Dr. Luna Michael from the VDL here helping with the initial coordination with the production systems, pathological findings, and laboratory findings, and also Dr. Marcelo Gottschalk from the University of Montreal and Dr. Anthony Vincent from the University of Laval in Quebec that we're working on genetic characterization of those strains. But like you said, Giovanni, in late November of last year, we started to see some activity of APP in the north central region of Iowa. And during approximately 60 days, we had 20 cases in a tightly geographical area of about 20 miles radius. And those were involved nine different production systems. And when we started digging in that, because the veterinarians were curious about how that was moving around in that region, we were able to identify a common serotype, which was APP serotype 15. And that triggered some more investigations, right? We contacted SCHIC, and then that led to the outbreak investigation that was led by Dr. Holtkamp and Machado. And what they learned there during those investigations is that the main factors that could lead to the lateral transmission of APP among those sites that were affected would be related to rendering practices and people movement. So biosecurity failures could be one of the main factors driving the lateral transmission of APP in that region during that time. So right now we are still seeing some cases. Right now we are at 30 cases involving at least 12 different systems. And what we are doing in addition to that initial outbreak investigation, we collected some samples from a site that had that broke to understand the post-outbreak dynamics in different sample types and also the environmental persistence of that APP to see if that could be actually one of the factors that's contributing to the transmission of the APP in the area. We are also doing the genetic characterization of those strains, like we mentioned with our collaborators in Canada. And what we learned at this point is that the strains involved in this outbreak, they are forming a distinct cluster within the serotype 15. So showing that's a very particular strain that's moving around. So what we're trying to understand now is there any thing different in these strains that's contributing to the outbreak the way we are seeing at this point? So we're keep digging on that, and we hope to share some more findings in the future. And the other thing that we're doing is also trying to understand the in vitro stability of this strain compared to the historical APP15 and also to a non-high virulence strain of serotype 1.
Those are great finds and lots of work there, Marcel. Thank you for doing that. And if I understood correct, there is two big take-homes there. The first one is we need to keep looking for genetic evolution of the agents over time. In this case, it seems like APP find a way to genetic evolve and be more pathogenic for this specific strain. And failures on biosecurity practices and biocontainment help it to spread the agent across different farms. So that highlights the importance to keep looking for how we do people and animal movement across farms, live or dead animals.
Mm-hmm. That's correct.
Marcelo, you mentioned that this new cluster for these outbreaks that occur. So all the outbreaks, the strains that you guys isolated, were within this cluster?
Yeah, there were two. There's at least two different clusters. So the biggest portion, probably 95% of the outbreaks were within this bigger cluster, and then we have a system that had outbreaks of APP15 that they were a little different than this main cluster. So that specific system, they were clustering with historical strains that we had in the US. So that might just be a coincidence that the outbreak was happening at the same time within the same region.
Makes sense.
And Marcelo, just another question. Do you think is that anything different that we can do just to improve the surveillance for APP and to avoid to have these cases again in the future?
I'm not sure if surveillance by itself, of course, it's very important to understand what's the status of your sow farm, to take measures, even if it's trying to eliminate or repopulate the sow farm with a clean breeding stock. But if not, at least understand that the downstream flow may suffer of that disease. And if that's the case, try to work with your neighbors and to increase your biosecurity practices in terms of biocontainment so the disease doesn't spread within the region.
Oh, that's amazing. Thank you for sharing this information. And just our last question right now. You worked for several years developing the family oral fluid sample type. So could you share with us more about the importance of these two for sow farm surveillance and how population-based sample for pathogen surveillance, how is important that to use this kind of tool for that?
Yeah, I guess in the last few years we've had the addition to our toolbox of many different sampling techniques that allowed us to understand the pathogen dynamics in the population. And that started with oral fluids, and then we had some evolutions including processing fluids, the family oral fluids now you guys are investigating tongue tips, and there are others that are out there, right? And what that allows us to do is to include more animals in every time we go do some sampling, that allows us to include some more animals or be more representative of the population, and then with that detect the pathogen of interest at lower prevalences, and with that usually we can also test less samples, so we can achieve that goal of at least monitoring for the same prevalence with fewer samples, or increase our probability of detecting a lower prevalence with also fewer samples, and that allows us, on the other hand, to also increase the frequency that we can do that sampling, right? And all that gives us the power to improve our surveillance systems by either detecting very, very low prevalences or working on early detection of those pathogens in herds that might be naive, right? And with that gives us, the veterinary, the chance to more information to work and to work with and make health-related decisions to the system, right?
No, very good discussion. Thanks a lot, Dr. Almeida, for bringing us this information, what happened in these previous APP outbreaks, what's happening currently with E. coli. And that was it for this month, guys. I hope you guys have enjoyed, and see you guys next month. Thank you.
Thank you.
Thank you very much.
Thank you.